Hello students, welcome to the EPG Pathshala. I am Dr. Rajeshwari Sinha, working as Program Officer at the Center for Science and Environment. Today, we are going to learn about vectors and restriction enzymes. Both vectors and restriction enzymes are essential components of molecular biology. This lecture will broadly have two sections. In the first section, we will learn what are vectors, their various properties, and what are the various different types of vectors such as plasmids, cosmids, bacteriophages, artificial chromosomes, shuttle vectors, and expression vectors. We will do a comparative understanding of these various types of vectors. Then we will move to understand what are restriction enzymes, how restriction enzymes work, and what are the various types of restriction enzymes. We will also see various areas where restriction enzymes can be used. Vectors are carriers of foreign DNA sequence from one organism to another organism. It replicates inside the host cell. The vector is an agent that can carry a DNA fragment into a host cell. If it is used for reproducing the DNA fragment, it is called the cloning vector. If it is used for expressing a certain gene in the DNA fragment, it is known as an expression vector. Essential properties of vectors: its own origin of replication called replicon, it should have a selectable marker, it should have unique restriction enzyme sites. Desirable properties of vectors: it should be short and small, compatible with host cell, incompatible with other vectors, must have high copy number, it should be able to express itself utilizing host machinery, it should be able to move under two systems, prokaryotic and eukaryotic systems. Plasmids are circular double-stranded DNA molecules that exist in bacteria and in the nuclei of some eukaryotic cells. They can replicate independently of the host cell. The size of the plasmid ranges from a few kb to a near 100 kb. Many types of plasmids are found in nature, in bacteria and in yeasts. They are circular DNA molecules, relatively small when compared to the host cell chromosome. They are maintained mostly in an extrachromosomal state. The nomenclature of plasmid. The name PBr322 conforms with the standard rules for vector nomenclature. P indicates that this is indeed a plasmid. Br identified the laboratory in which the vector was originally constructed. Br stands for Bolivar and Rodrigues, the two researchers who developed PBr322. 322 distinguishes this plasmid from others developed in the same laboratory. There are also plasmids called PBr325, PBr327, etc. The structure alongside shows an E. coli plasmid cloning vector PBr322. The PUC plasmids. PUC plasmids stand for plasmids developed at University of California. PUC plasmid was constructed by J. Messing and J. Vieira. This plasmid contains 40% DNA similar to PBr322 and has ampicillin resistance gene. It has a short stretch of DNA with a cluster of restriction enzyme recognition sites. This stretch is called as multiple cloning site, abbreviated as MCS. PUC18, PUC19. These vectors are small, high copy number E. coli plasmids of 2686 base pairs in length. They are identical except that they contain multiple cloning sites MCS arranged in opposite orientations. PUC 18 and 19 plasmids contain the following: the PMB1 replicon REP responsible for the replication of plasmid, the BLA gene coding for beta lactamase that confers resistance to ampicillin, region of E. coli operon LAC containing cap protein binding site, promoter P LAC, LAC oppressor binding site, and 5' terminal part of the LAC-Z encoding the N terminal fragment of beta galactosidase. The figure shows the structure of PUC18 and 19 plasmid cloning vector. Now we will see what is a Cosmid. A Cosmid, first described by Collins and Horn in 1978, is a type of hybrid plasmid often used as a cloning vector that contains Cos sequences, DNA sequences originally from the lambda phage. Cosmids can be used to build genomic libraries. The Cosmid vector is a combination of the plasmid vector and the Cos site, which allows the target DNA to be inserted into the head. Cosmids are able to contain 37 to 52 kb of DNA, while normal plasmids are able to carry only 1 to 20 kb. They can replicate as plasmids if they have suitable origin of replication. Advantages: high transformation efficiency. The Cosmid vector can carry up to 45 kb, whereas plasmid and lambda phage vectors are limited to 25 kb. This figure shows cloning by using Cosmid vectors. The figure A shows that in addition to the AMP, ORI and polylinker as in the plasmid vector, the Cosmid vector also contains a COS site. Part B shows after Cosmid vectors are cleaved with restriction enzymes, they are ligated with DNA fragments. The subsequent assembly and transformation steps are the same as cloning with lambda phages. What is a bacteriophage? In the 1940s, Max Delbruck and the phage group that he brought into existence laid the foundation of modern molecular biology by studying bacteriophage. The term bacteriophage is often called phase and can be used to describe either one or many particles of the same type. The plural term phases is used when different types of phase are being considered. Bacteriophage are viruses that attack bacteria. Most phage infect the bacterial cells they infect, lytic phages, but many other phage follow lysogenic cycle. In this case, phage chromosome integrates into the bacterial chromosome. The most commonly used E. coli phages are lambda phage and M13 vector. In this slide, we will look at the lytic cycle and the lysogenic cycle. In step 1 and 2, we see the adsorption and penetration, where phage is adsorbed to receptor site on bacterial cell wall, penetrates it and inserts its DNA. In the lytic cycle, the biosynthesis step takes place. The phage DNA directs the cell's metabolism to produce viral components such as proteins and copies of phage DNA. Empty phage heads and pieces of phage DNA. In step 4, maturation takes place and the heads are packed with DNA. In step 5, the bacterial cell lyses, releasing completed infective phages. In the lysogenic cycle, after the adsorption and penetration, the phage DNA inserts itself as a prophage into the bacterial chromosome. The phage replicates along with the bacterial DNA prior to binary fission. Indefinite cell divisions take place, the binary fission is completed and each cell has a phage DNA incorporated. After induction, the prophage forms. Phage vectors present two advantages over plasmid vectors. They are more efficient than plasmids for cloning of large DNA fragments. The largest cloned insert in lambda phage is 24 kb, while for plasmid vector it is less than 15 kb. It is easier to screen large number of phage plaques than bacterial colonies for identification of recombinant vectors. Lambda phage vector. The genome size of the lambda phage vector is 48,502 base pairs. It contains an origin of replication, a gene for head and tail protein, the enzyme for DNA replication, lysis and lysogeny. It is single-stranded, protruding cohesive end of 12 bases, the other end is complementary. Lambda genome remains linear in phage head, but in E. coli the two cohesive ends anneal to form circular molecule. The sealed cohesive end is called a Cos site. The gene for lysogeny are located in the segment between the 20 to 38 kb. The whole or part of it are deleted to create a lambda vector for accommodation of large DNA inserts and to ensure that the recombinant phage is always lytic. Phages are viruses that can infect bacteria. The major advantage of the phage vector is its high transformation efficiency, about 1000 times more efficient than the plasmid vector. The figure alongside shows schematic drawing of the DNA cloning using phages as vectors. The DNA to be cloned is first inserted into the DNA replacing a non-essential region. Then, by an in vitro assembly system, the viron carrying the recombinant DNA can be formed. The genome is 49 kb in length, which can carry up to 25 kb foreign DNA. M13 vector. M13 vectors are used for obtaining single strand copies of cloned DNA which are suited for DNA sequencing and in vitro mutagenesis. They are derived from the 6.4 kb genome of the E. coli filamentous bacteriophage M13. They carry several genes specific for phage DNA replication and gene coding for the component of phage protein coat. Properties of M13 vectors: very large inserts and can be cloned. Pure single stranded copies of DNA of double stranded DNA inserts are obtained. They form plaques like lambda phage, making selection of the recombinant vector easy. Artificial chromosomes, developed by David Burke et al. In the year 1987. The essential components of yeast artificial chromosome vectors. These contain centromeres, telomeres and autonomous replicating sequence for proliferation in the host cell. The AMP for selective amplification and markers such as TRP1 and URA3 for identifying cells containing the YAC vector. Recognition sites of restriction enzymes, for example EcoR1 and BAMH1. Yeast artificial chromosome. The YAC vector is capable of carrying a large DNA fragment up to 2 MB, but its transformation efficiency is very low. The bacterial artificial chromosome. BAC vectors are similar to standard E. coli plasmid vectors. They contain the origin and genes encoding the ORI binding proteins required for plasmid replication. They are derived from a naturally occurring large plasmid, the F factor, the low copy number, low transformation efficiency overcome by electroporating recombinant DNA into E. coli. Advantages of bacterial artificial chromosomes over yeast artificial chromosomes: high stability, ease of transformation, speed of growth of E. coli host, simpler to purify, more user friendly. P1 derived artificial chromosome PAC. This cloning system was developed by Loanu et al. In 1994. The constructed vector incorporates features of both P1 and F factor systems and can be transformed into E. coli by electroporation. In a PAC vector, inserts of size 100 to 300 kb can be cloned. It is devoid of problems such as chimerism and instability of cloned DNA. In this slide, we have a snapshot of the comparison of vectors. The various vectors such as plasmids, phage, cosmids and phasmids are compared in terms of origin, composition, feature, usage, shortcoming, example and attention. For example, in terms of origin, the plasmid vector is derived from bacteria, phage is derived from double stranded DNA virus, cosmid is derived from plasmid plus cos site of lambda phage and phasmid is derived from plasmid and phage. In terms of composition, plasmid is a double stranded DNA ORI region, phage has a cos site and origins of viral and complementary strands, cosmid has a plasmid replicon and a cos site of lambda DNA, and phasmid has plasmid origins and phage origins of replication. The choice of vector. This slide shows the targeting fragment length for each kind of vector. For example, the plasmid can have a target fragment length of 0 to 10 kb. A replacement lambda vector can have a fragment length of 9 to 23 kb. Insertion lambda vector can have a length of 0 to 10 kb, cosmids 33 to 44 kb, PAC 130 to 150 kb, BAC 300 kb and YAC 0.2 to 2 MB. Shuttle vector. A shuttle vector is a vector, usually a plasmid, constructed so that it can propagate in two different host species. Therefore, DNA inserted into a shuttle vector can be tested or manipulated in two different cell types. The main advantage of these vectors is that they can be manipulated in E. coli and then used in a system which is more difficult or slower to use, example yeast or other bacteria. Properties. The vector must replicate in many organisms, example bacteria, yeast and plants, to facilitate the isolation and characterization of gene. The vectors must be easily recognized by selectable markers. They should be small in size to accommodate DNA inserts. Cloned genes should be easily detected. Useful quantities of vector must be easily obtained. The vector must be stable, non-pathogenic and non-stress inducing. Expression vector. An expression vector, otherwise known as an expression construct, is generally a plasmid that is used to introduce and express a specific gene into a target cell. Expression vector allows production of large amounts of stable mRNA. Once the expression vector is inside the cell, the protein that is encoded by the gene is produced by the cellular transcription and translation machinery. The plasmid is engineered such that it contains a highly active promoter which causes the production of large amounts of mRNA. Features of expression vector: origin of replication that is functional in target host cell, antibiotic selection gene or other gene selection mechanisms, promoter strong or weak as per suitability along with regulatory control, restriction sites immediately downstream from the promoter. The TI plasmid. Features of the TI plasmid are as follows. The tDNA contains oncogenes and opine synthesis genes and it is transferred into the host plant genome. The VIR region regulates the transfer of tDNA into plant cells. Opine catabolism regions produce enzymes necessary for the utilization of opines by agrobacterium. The TRA region, conjugative transfer, functions in conjugative transfer of the plasmid. Origin of replication for propagation in agrobacterium. We have now reached midway into this lecture and just finished learning about vectors. We saw different types of vectors in detail and also some of their specific examples. Now, we are going to look at what are restriction enzymes. These are enzymes which are used to cut DNA. We will look at a brief history of these enzymes, how they work, their nomenclature, the 4 different types of restriction enzymes and the reaction conditions for digestion of restriction enzymes. At the end, we are also going to look at the concept of restriction mapping and the application of restriction enzymes. Now, we will look at what are restriction enzymes. Restriction enzymes are the enzymes that cut the DNA at specific sequences producing either blunt or sticky ends. Due to their mechanism, they are rightly named as molecular scissors. This is naturally occurring phenomenon in bacteria. We can say it is bacterial immune system or the host control restriction and modification of foreign DNA. Most of the bacteria use this mechanism to prevent its DNA from foreign invading DNA brought out by viruses. DNA which is unable to cope with the host modification is sure to be degraded. History of restriction enzymes. H. O. Smith, K. W. Wilcox and T. J. Kelly were the first who isolated the useful enzyme in 1968. In 1969, Stuart Lynn and Werner Arber discovered the enzymes which cut DNA at specific sites. How restriction enzymes work. Restriction enzymes are discrete class of endonuclease enzymes that hydrolyze the phosphodiester bonds within the DNA strands. This can be prevented if the DNA is being methylated. Restriction sites are mostly palindromic sequences. These enzymes work by scanning the entire DNA and then cuts it at the specific cleavage site meant for it. These are highly site specific in nature. This slide shows some of the restriction enzymes such as HAE3, BAMH1, HINT3, EcoR1. HAE3 and ALU1 are restriction enzymes that produce blunt ends whereas BAM-H1, Hint3 and EcoR1 are restriction enzymes which produce sticky ends. Nomenclature of restriction enzymes. Names of all restriction enzymes are derived from genus and species name. The first letter is for the genus of the bacterium, the next two letters are for species of bacterium, the fourth letter is for strain designation if needed. The name ends with Roman numeral depending on the number of such enzymes occurring in that organism. This table shows the scheme of RE nomenclature as discussed in the previous slide. For example, the name of the restriction enzyme EcoR1 is derived as follows. The alphabet E is taken from the genus Escherichia, the alphabet CO taken from the species coli, and the alphabet R taken from the strain RY13. Similarly, for BAM-H1, the alphabet B is taken from genus Bacillus, the alphabet AM is taken from species Amyloliquefaciens and the strain H. 4, 6 and 8 base pair cutters. How often will a 4 base pair cutter cut a piece of DNA? The answer is 1 every 256 base pairs. How often will a 6 base pair cutter cut a piece of DNA? It is 1 in every 4096 base pairs. How often a 8 base pair cutter cut a piece of DNA is 1 in every 65,000 base pairs. Types of restriction enzymes. There are 4 main types of restriction enzymes, types 1, 2, 3, 4. Type 1 restriction enzyme hydrolyzes DNA thousands of bases away from their recognition site. Type 3 restriction enzymes cleaves the DNA outside their recognition sites, so require 2 identical sequences in opposite orientation, thus can do the restriction and modification activities. Type 4 restriction enzymes combine with a type 2 restriction enzyme to form the modification activity. Type 1, 3 and 4 are not used for recombinant DNA technology. Type 2 restriction enzymes that are used mostly because it cleaves most sites within sequences. Isoscizomers. All the different restriction enzymes which possess identical recognition sequences are called isoscizomers. They share the same cleavage and recognition sites. There are few enzymes which differ in their recognition sequences but still generate identical overlapping or flanking regions at the termini. Reactions, conditions for restriction enzyme digestion. Factors influencing the restriction enzyme activity include ionic strength, pH, temperature and monovalent cations. Most restriction enzymes are active at 37 degrees Celsius, though thermophilic organisms require higher temperature for maximal activity. Here we look at what is restriction mapping. A given DNA fragment will generate a characteristic pattern of restriction fragments known as a restriction map. If the DNA sequence is known, then restriction map may be predicted. There are many commercial programs available to predict restriction fragments. These include web cutter, neb cutter, restriction mapper, etc. This slide shows lambda Hint3 restriction map. This slide basically compares type 1, type 2 and type 3 restriction enzymes in terms of parameters such as protein structure, recognition sites, cleavage sites, restriction and methylation and ATP consumption. For example, the type 1 enzymes have a bifunctional enzyme of 3 type of subunits. Type 2 enzymes have separate endonuclease and methylase 2 subunit types. Type 3 enzymes are bifunctional enzymes with 2 subunits. The recognition sites in type 1 enzymes are bipartite and asymmetrical, that in type 2 enzymes are short, 4, 6 or 8 base pairs long, usually palindromic, and that in type 3 enzymes are asymmetrical with 5 to 7 base pairs. Cleavage sites for type 1 enzymes are non-specific, greater than 1000 base pairs away from the recognition site. Cleavage sites in type 2 enzymes are same as that of the recognition site, while cleavage sites in type 3 enzymes are 24 to 26 base pairs downstream of the recognition site. Restriction and methylation. In type 1 enzymes, some enzymes catalyze both reactions. Two separate reactions of restriction and methylation are carried out by type 2 enzymes, and some enzymes catalyze both reactions in case of type 3 enzymes. ATP consumption exists in case of type 1 and type 3, but not in case of type 2 enzymes. Applications of restriction enzymes. They are used to cut a DNA into small fragments for the isolation of the gene of interest. They are used to cut out unwanted sequences from natural vector DNAs to construct activated vectors. They can be used to cut vectors' DNA's well-defined sites for cloning purpose. They are mainly used in DNA sequencing. They help in restriction mapping and prediction of gene loci. Restriction enzymes are used in preparation of molecular markers like RFLP, RAPD, AFLP, etc. They are mainly used in DNA sequencing and help in restriction mapping and prediction of gene loci. They are also used in the preparation of molecular markers. Students, we have now come to the end of this lecture. Today, we learnt about vectors and restriction enzymes. Let us summarize briefly what we learnt today. Vectors are agents that are used to transfer specific sections of the DNA from one microorganism to another, the host cell. Vectors are of different types. They can be plasmids, cosmids, bacteriophages, artificial chromosomes, shuttle vectors, expression vectors, and the TI plasmid. An expression vector, also known as an expression construct, is generally a plasmid that is used to introduce and express a specific gene into a target cell. Restriction enzymes are endonuclease enzymes that cut the DNA at specific sequences, producing either blunt or sticky ends. These are used to isolate the gene of interest. There are four main types of restriction enzymes: type 1, type 2, type 3 and type 4. Factors influencing the restriction enzyme activity are ionic strength, pH, temperature and monovalent cations. Thank you for watching this lecture on vectors and restriction enzymes.